Okay, folks at home, somebody give me a heads up, like, can you hear me? Yeah, I'm not here. Can you hear me?
We can hear you.
Okay,
Yes.
good. Anthony, I have a lab and an exam. Trying to give back labs and exams if you've turned them in on paper, so please, just. All right, and although some of you may not want to see your second exam because it was a low average, it's still yours, not mine. So I appreciate it if you made arrangements to come and get them. Okay, so today's topic is going to be parasites and their hosts as a variation on the basic exploitation system. So what we've covered so far is exploitation in nature, like the challenges, what we have to describe. We've developed some models that describe those things and allow us to predict the future when we see these interactions. And then we've asked how evolution affects these interactions. And we saw what happens when predators get more efficient, and basically it's an arms race. And we've seen what happens when prey get more efficient at using their resources, which can lead to the paradox of enrichment. But now, it's important to understand that the way that parasites and predators and parasitoids work is much more diverse than the way that competitors work. So if you think about it, 2 of the 3 kinds of exploiters have to kill their victims. And that's parasitoids and predators. In each case, once the exploiter kills its victim and consumes it, then it is free to fly off or run off or however, and find another. So they're kind of free agents. They are able to leave the host once they've killed it and consumed it and search for another. All right, well, so they don't permanently dwell within their victim, but that's not the same as parasites. Parasites are fundamentally different because they dwell in or on the victim for their entire lives, usually, or most of their entire life. So there are some special features of parasites that differ from your classic predator-prey system of lions killing zebras. So today, we'll first discuss parasites and their hosts as general exploiter-victim systems, okay? And we'll talk about what parasite virulence is and how it's related to efficiency of a predator. And we'll talk about some mechanisms of host resistance. We will then give you an example of a parasite-host interaction, which is just absolutely classic as a demonstration of the Red Queen in operation. The perpetual arms race between exploiters and their victims. Every time the exploiters get an advantage, they select on the victims, and the victims that survive have an inborn advantage against that predator. And so the cycle never ends. Then we will look at some special features that we need to model for host parasite systems. And this is the basis of medical epidemiology. So it's quite different from the kinds of host parasite or predator-prey systems we've been modeling, there's a fundamental difference in the way we view hosts and parasites that influences the way we develop these models. So then we'll look at the virulence of parasites and how that is related to something called the transmission barrier. And finish up using complex parasite life cycles as a solution to the limited availability of hosts at times. So first off, how do we frame parasites and their hosts is in the terms of a general exploiter victim system. And I guess I got to go down here and get my advanced. There we go. All right. Well, so first let's consider the features of parasites. They're typically smaller than their hosts. All right. Predators are quite large compared to their prey. Parasitoids are small relative to their hosts, but they kill their hosts. Parasites are smaller than their hosts. So they can be macroscopic like tapeworms or insects or leeches. They can be microscopic like bacteria and viruses. They can be external or internal. You can have external parasites like ticks, fleas, caterpillars, if you're a plant. If you're a human, you're unlikely to have caterpillars as an external parasite. However, it does happen in at least one case. They're also mostly internal. That's the way we think of them anyway. That's like tapeworms, bacteria, viruses. They all live internally, sometimes within an organ, sometimes within the cells themselves. Now, the fundamental feature of parasites is that they steal resources in order to reproduce. They do not care about killing their host. They simply want to steal resources. If they do cause the death of the host, that's a complete accident. And it harms the parasites because when the host dies, so do the parasites. It's less true of external parasites, but it is very true of internal parasites. Okay, and so for these reasons, there are differences in the way that parasites interact with their hosts compared to the way predators and parasitoids interact with their prey. So let's just slap up a predator-prey graph up here and change it to be an exploiter, a parasite host graph, and let's examine what would be the same and what would be different. Okay, well, we know that over time, evolution makes predators more efficient at using their hosts, because the more efficient a predator is, the more offspring it leaves compared to less efficient predators. Well, it's the same thing with parasites. Over time, evolution will favor parasites that are better at stealing resources from their hosts within the host. So if you get a virus, natural selection will act to make that virus more efficient at making copies of itself, stealing your resources. And the strain of virus that is most efficient is the one that will come to dominate your body while you're sick. So more efficient parasites reproduce faster because they're able to steal more resources, and they're favored by natural selection. So that's exactly analogous to coyotes getting faster when they're chasing jackrabbits, and the predator isocline will move to the left as a result. Okay, now here's the problem. That if you are more efficient at stealing resources from your host, you're making the host sicker. And you are raising the likelihood that that host will die. So that right there is a problem that predators and parasitoids don't have. They need to kill their host or victim. Parasites don't. So in terms of epidemiology, when we talk about parasites, this efficiency, the ability to cause sickness or death, is called virulence. And so the equivalent of becoming a faster or smarter predator is becoming a more virulent parasite. You become more virulent because natural selection favors parasites that steal resources from their hosts at a higher rate and make more copies of themselves. So virulence is what we're looking at in parasites. And we will call it virulence instead of efficiency. Okay, so natural selection then inside the host will cause parasites to make you sicker and sicker and sicker over time. So why don't we all die? Well, obviously because when an exploiter makes exploits a victim, the victims that are most effective at countering the way that it's exploiting them will leave more offspring. And it's the same thing with parasites and their hosts. So host resistance is the countermeasure that natural selection produces in the host. So a parasite may become more virulent over time, but at the same time, it is then selecting for the hosts that have the best ability to resist the parasite. So host resistance plays the same role in host-parasite interactions that selection on a jackrabbit might play in a coyote-jackrabbit interaction. If coyotes get faster, they catch the slow jackrabbits and only the fast ones are left. If parasites get better at stealing resources, they kill off the most susceptible hosts and only the ones that have the greatest resistance to the parasite are left. So once again, the Red Queen is in operation here. This kind of interaction is running as fast as they can to stay in the same place. If the parasites get an advantage over the hosts and the hosts cannot respond, the parasites could cause the extinction of the host. If host resistance outruns the ability of parasites to adapt, the host could cause the extinction of the parasite. Okay. Now, host resistance is generally in the immune system, and it is found in all advanced organisms in some way or another. We're used to talking about the human immune system, but it doesn't matter what you're talking about, there are immune responses. Yes, question in class.
Why is the parasite like taking the why would not selection choosing the factors that I guess virulence or ultimately
well, parasites go through generations. So the question is, why would natural selection push parasite virulence to the point where it could potentially kill the host? Well, parasites have generations just like everybody else. And who is represented in the next generation? Well, it's the form that has the highest reproductive rate. So if one parasite can produce 5 copies of itself in a single parasite generation, but another one can produce 10 copies, then over time, the parasite population in your body will come to resemble the one that can produce greater numbers of itself. And it is completely irrelevant what the outcome is for the host. Natural selection favors the individual. It does not favor the population. What is good for individuals is not necessarily good for populations. Natural selection can cause parasites to become so efficient that they cause their own extinction. It's just the way it is. Okay. So in animals, we then have the immune system of some sort. Plants have physical and chemical defenses, and we will talk about them Wednesday. But in either case, the evolution of host resistance prevents parasites from gaining too big an advantage. It stabilizes the interaction. So the Red Queen, where we have this endless arms race between parasite and host, is exactly the same arms race that we have between predators and prey, between parasitoids and their hosts. Now, here is one of the classic illustrations of the parasite-host interaction that shows the Red Queen in operation, right? So here is a kind of pictogram of rust wheat, or wheat rust, which is a parasite, invading populations of wheat in the American Midwest between 1956 and about 1970. Okay, and what you see here is that there are different strains of wheat rust. The strain 17 accounts for about maybe 15% of all infections in 1956. Strain 17 accounts for maybe 25% of all infections in 1956. But look what happens over time. Strain 10 basically goes extinct by 1960. Why? Because wheat plants had evolved resistance to strain 10. Strain 11 also basically went extinct a little longer, but about 1967, strain 11 went extinct because wheat plants gained complete resistance to it. Okay, strain 9 originated about 1956. It never really got much traction because wheat plants were already kind of resistant and they rapidly caused its extinction. Okay, but what you see here is strain 17 started out at about 25% of the infections in the wheat populations that were being sampled, and rapidly expanded to be about 2/3 of all infections. So strain 17 took over because it was better able to exploit the wheat plants than strains 10, 11, 9, or 15, which all declined. Okay. However, eventually, look what happened to strain 17. It went extinct about 1969. Because it made up most of the infections. That means that most of the plants that were infected with strain 17, and some of them survived. The plants that survived passed on their genes to the next generation in the next year. And because they were resistant to strain 17, the wheat population became more and more and more like the resistant plants. And eventually, the entire population became resistant to strain 17, and it went extinct. But that's not the end of the story, because we had a new strain, strain 18, originate in 1961, and it rapidly took over and accounted for about 80% of all infections by 1970. What you see is the parasite gaining an advantage and expanding its grip on the host population. And you see the host population responding with the evolution of resistance. So strain 18 may have accounted for 80% of the infections in 1969, but come 1975, strain 18 probably would be extinct, and a new strain of wheat rust would have taken its place. Question in class.
Some of the decline of strain 17 around 1962 partially due to strain 18 outbreeding. So it's not just that the wheat has become resistant, but it's just strain 18 is better.
Okay, so the question then is, strain 17 starts to decline about 62, and that's when strain 18 arises. Is it possible that competition between those two strains accounts for at least some of the decline in strain 17? It is possible, but in wheat rust infections, it's typically a single strain that is infecting a particular wheat plant. So there's not the ability to compete within a wheat plant for dominance between these two strains very often. What it's more likely is who's able to colonize a wheat plant and become the only strain that's infecting the wheat plant. So it is possible that some of this is due to direct competition, but it looks like most of it is probably due to the evolution of host resistance. So it's a little bit different than in, say, free agents like parasitoids and predators, where they can roam around and actually directly compete. It's not as easy for parasite strains to directly compete. However, it is possible. And we have investigated this in some detail in the laboratory, and I'll summarize some of those results a little bit later. When we do have strains within a host competing, really interesting things happen. Okay, so how do we model these things? Well, all right, for very selfish reasons, we don't care about the number of parasites the way we do about the number of predators or parasitoids. All of our models of parasite-host interactions are based on human discomfort. And so we don't care how many parasites there are, we just care how many infections there are. And let's see if we can't avoid getting an infection, regardless of whether it's one parasite or a million parasites. So a fundamental difference between the way we model parasites and hosts, and that includes all diseases, and predator-prey or parasitoid host interactions, is that we don't model the number of parasites. We model the number of infections. So we organize our models fundamentally differently. And the basic model for all epidemiology is the SIR model, the susceptible, infected, and resistant model. So basically it has three things. It has three numbers and two rates. It has S, the number of individuals who are susceptible to a pathogen. And you can express this as actual numbers. You can express it as percent of a population. There are slightly different ways of doing the calculations, but they wind up being equivalent. I is the number of individuals that are infected. And so I'm going to stick with calling it the number of individuals. And then R is the number of individuals that develop resistance. And we're going to assume that individuals are not dying. We're going to assume that this is a non-lethal parasite-host interaction. And the only three things that can happen is that you can become susceptible, or you can be susceptible, you can become infected, and once infected, you can recover. So clearly, you can stay infected for a long time before you recover, or you can recover very quickly. Well, those things will affect the course of a disease outbreak. The two rates that we're interested in are G, which is the rate of transmission between individuals. So you have a susceptible individual come in contact with an infected individual. What's the probability that the infection will be passed? Generally, it's pretty low, even for very virulent diseases, but it's non-zero. And then B is the rate of recovery and development of immunity. So once you're infected, B is the rate at which you get over your infection. And we're going to assume one other thing. Not only is it non-lethal, but once you are recovered, you have complete immunity. That's the simplest way to view this. So if we view a non-lethal infection that starts in a population and there's some recovery that confers complete resistance, what does the infection look like? Okay, we need to do our, make sure that we understand how we're going to get the numbers we're going to graph. So the probability of contact between infected and susceptible individuals is simply S times I. And I think you can see if you're talking about like number of susceptible and number of infected, you'll have to have a different way of calculating it than if you've got like proportion of the population that's susceptible and proportion that's infected. We're actually not going to deal with that. We're just going to say S and I. And we're going to leave those calculations out of this. You're not going to be asked to calculate any of this. Okay, the rate of infection, the rate of new infections is then the rate at which susceptible and infected individuals come together times G, the rate of transmission. Okay, so that's the birth rate. S times I times G is the birth rate of new infection. And then the rate of recovery is I, which is the number of infected individuals, times B, the rate at which they recover. So that's equivalent to the death rate. So just as in all of our kind of population models, we have a birth rate and a death rate. S times I times G is the birth rate of new infections. I times B is the death rate of infection. And now that we have this, we can calculate the famous R0, or R0, as the British call it, and kind of has taken over the COVID literature. But R sub 0 is exactly equivalent to the thing we talked about in the life table. It is the number of infections that a single infected person can expect to create prior to getting better. Okay, so the higher that number, the more transmissible the disease is. Okay, so that's births divided by deaths. Well, clearly, if births are greater than deaths, then each infection will cause more than one infection, and the disease will spread. If R sub 0 is less than 1, then each infection results in less than one infection. And so when that person gets better, it will have transmitted less than one infection to a new person, and the disease will decline. Now, over time, remember, you start out with a ton of susceptible individuals. So the birth rate of new infection should be pretty high. But over time, you get mostly already infected or recovered individuals. So the birth rate should drop. So when we make a graph of how these things should work, the population starts 100% susceptible. Okay, and as soon as individuals start getting infected, some of the susceptible individuals are lost because they're now infected, right? And so eventually that drops quite fast. Here is the number of infected individuals, and that increases up to this point. But then the number of infected individuals starts to decline because it gets harder and harder to find a new host. And that's because some of these infected individuals are recovering and can't be reinfected. So at this point right here, this is the inflection point that changes from a spreading disease to a declining disease. So R sub 0 is greater than 1 here, where you have lots of new uninfected hosts. It reaches a peak and then starts to decline as the number of recovered individuals starts to greatly outweigh the number of susceptible individuals. So that peak in infections signifies the change between R0 greater than 1 and R0 less than 1. R0 is greater than 1 here, which explains why this curve is increasing. But now it's declining. And it's declining because each individual infects less than one individual, because there are so many fewer susceptible individuals left. So R0 is less than 1 here. And eventually, the disease outbreak dies out. Okay, this looks like, kind of a relatively simple cartoon, but it actually is the basis for modeling infections like measles outbreaks and influenza outbreaks. It's pretty much the basis for all of our understanding of how disease transmission works in humans. Well, not just humans, pretty much everybody. So what's important here? What's important is to understand what is the birth rate. It's S times I times G. What's the death rate of the infection? It's I times B. And what is R0? It's the birth rate divided by the death rate. Birth rate divided by the death rate greater than 1, the infection spreads. Birth rate divided by death rate is less than 1, the infection dies out. Okay, and it's then the other thing I want you to really focus on is the importance of this inflection point. Why does it happen? Because susceptible individuals are crashing, resistant individuals are skyrocketing. It becomes harder and harder to find a new host. And so R0 changes from greater than 1 to less than 1. So although there's a lot of terms here, we're not going to actually do any calculations with this model. I simply want you to understand the components of the model. So this basically, if you go to the Tulane School of Public Health and study epidemiology, you get a whole bunch of this. And of course, this is day 1 for them. And then they go into enormously complicated variations on these models. But let's switch gears now and talk about a little more ecological stuff, which is the transmission variant and what it has to do with the virulence of parasites. And this gets at a question that we just had. Why would natural selection push parasites to be virulent enough to cause the death of their host? Well, that's related in some way to what we're going to talk about now. So remember that parasites have different dynamics than parasitoids and predators. Parasitoids and predators have to kill their hosts, and then they have to move on to find another host. But parasites live on or in the host. And it's hard for them to find a new host. They can't go actively search the way that predators and parasitoids can. They have to rely on their host coming in contact with another host. So that's harder. They are not free agents here. And if their host dies before they can find a new one and ensure transmission, the parasite dies, and its fitness is 0. So there is a transmission barrier in parasites that parasitoids and predators do not have. Parasites are stuck with this quandary. They want to reproduce themselves in a host, but they can make the host sick and die. And the end product is they have to get out of that host before that happens. So they want to compete within the host, but they also want to survive long enough to get to a new host. That's not a problem that parasitoids and predators have. It is unique to parasites. So this transmission barrier is really important in our understanding of the ecological aspects of these interactions. So what that then means is that parasites face a trade-off between virulence, which is the ability to make a lot of copies of yourself really fast and win competition inside a host, versus the ability to be transmitted to another host. If you're highly virulent, you reproduce rapidly, you make lots of copies, you steal lots of host resources, and you run the risk of killing a host. But you win the competition inside the host. However, doing that could ensure your extinction. So there's a trade-off between success within a host, which is relying on virulence, the ability to make lots of copies of yourself really fast, and transmission, which is the ability to find a new host. The longer your host lives, the more likely transmission is. But that's completely at odds with how natural selection pushes parasites to be more efficient within the host. So there are opposing selection pressures now. Parasites get higher fitness from succeeding within the host, but they get higher fitness from being transmitted ultimately. And those two things are in opposition. And that's not a problem that predators or parasitoids have. So what determines then the balance between virulence and transmission? It's the availability of new and uninfected hosts. If uninfected hosts are common, you can have a highly virulent parasite. Because even if it kills its host rapidly, there's a lot of opportunities to find a new host. Okay, so when uninfected hosts are really common, virulence can have an advantage. You make lots of copies of yourself. And because there are lots of uninfected hosts, it's easy to find a new host, regardless if you kill the one you're in. So when uninfected hosts are common, virulent strains have an advantage. But if new uninfected hosts are rare, highly virulent strains simply kill their hosts and never get transmitted. And natural selection favors the evolution of lower virulence. Low virulent strains have higher fitness when uninfected hosts are rare. So depending on how easy it is to find a new host, you can support high virulence, or you may be forced to evolve lower virulence. That's not the way predators and parasitoids work. The pressure there is always for greater efficiency. So let's look at that graph again and see what the consequences are for virulence. All right here, there are lots of susceptible individuals. It's okay for this parasite to be virulent. It can be really, really efficient. It can make you very sick. But over here, there's very few new hosts available. You make your host very sick, it will die before you get transmitted. So as time goes on, pretty much all parasites can be forced to evolve lower virulence. They start out highly virulent in a new uninfected population where everybody's vulnerable. But as vulnerable individuals become rare, virulence has to decline. And this is really interesting. It doesn't matter if there's evolution of host resistance. It's just the sheer numbers of uninfected hosts. Parasites evolve lower virulence over time, regardless of whether their hosts can evolve resistance. So let's look at an example here, classic example, which is the disease biology of syphilis. Now, syphilis is a member of a family that includes disease-causing organisms that include yaws and pinto. There's something called endemic syphilis, which is not easily transmissible. It's a skin disease. And basically, all of these diseases infect epidermis. The innovation that syphilis has is that it infects mucosal membranes rather than epidermal skin, and is easier to transmit because of that. Now, there are all kinds of theories about how syphilis evolved. There are certainly traces of what looks like syphilis in skeletons from London from the year 960, I think. But there's no molecular evidence that that was what we think of as modern syphilis.
Mohammed,
One of these
are
other
you
things.
bringing me to work?
I think we need to mute somebody at home.
My dad is cutting grass. He can't. Who's gonna bring me?
Let's mute everybody at home. Okay. The first
What time
outbreak
are
of
you going to
modern,
work for?
let's mute everybody. Well, all right, keep it down out there. The first recorded outbreak of what we think of as modern syphilis occurred at the Siege of Naples in 1495. And of course, it's really suspicious that that was just after Columbus came back from the New World. The likelihood seems to be that, yes, it was picked up in the New World and transmitted back to Europe. But it was an incredibly virulent disease. It was a horrible disease that killed very rapidly, within weeks when it first outbroke. That doesn't sound like syphilis that we know. But once people understood that it was transmitted by sex, they started taking precautions, started using condoms to reduce the rate of transmission. And those lower transmission rates have led to the evolution of a less virulent syphilis disease, the one that we know today, that is a slow burning infection that takes 20, 30 years to emerge as a killer in people because it is now so hard to transmit. But in a naive population where everyone was susceptible and there was no transmission barrier, syphilis was incredibly virulent. It killed a lot like Ebola kills today. But it doesn't now because of the transmission barrier. It's much harder to transmit now than it used to be. Ergo, you cannot support high virulence. You must evolve lower virulence. Oops, and let's see, we gotta get down here into this. Okay, so there's an evolutionary take-home message here. Coevolution between parasites and their hosts produces progressively lower virulence, even without the evolution of host resistance. Simply having fewer uninfected hosts forces parasites to become less virulent. So even if the hosts all die, it doesn't matter. The parasite will become less virulent over time. Now that causes less harm to the host. It causes more benefit for the parasite because it allows it to be transmitted. If this goes on indefinitely, it could even produce a commensal rather than a parasitic relationship in which one organism lives on another without harming it in any way, either or positively affecting it. Commensals live in their hosts, but don't have any effect on their fitness. It's regarded as a rare interaction in ecology, but that's probably because we just haven't identified all of them. But there's at least a possibility that it could even eventually become mutualistic if evolution goes on long enough. Because remember, anything that benefits one of the parties will be selected for. If something about the parasite or former parasite benefits both the parasite and the host, well, hey, that will be rapidly selected for. Yes. By the is that something like the presence of E. coli in our guts? The answer is yes. That's exactly the kind of thing we're thinking of here. Okay, so that's the evolutionary take-home. What about the public health take-home message for you guys sitting out there? Any action that you take that increases the transmission barrier of a disease of humans will reduce the rate of transmission and produce lower virulence in the things that afflict us. That's regardless of whether you have any resistance or not. Now, in the HIV epidemic, HIV was lethal within about 6 months in the 1980s. It's not lethal today. And that's not just because we have drugs. It's because people figured out very quickly that it was transmitted by sex. They started using condoms. They reduced the transmission rate. HIV had no alternative but to become a chronic rather than acute infection. You can directly affect the virulence of the diseases that affect you. Okay? And regardless of the political stuff that's going on out there, masks reduce the transmission of COVID-19. You select for less virulent strains of COVID-19 when you reduce the rate of transmission. Those who do get sick will get less sick because of the transmission barrier. And people have control over how sick they get. Problem is that it's not necessarily easily visible to individuals. It is a population thing that has to happen. Transmission at the population level is the key. And people don't relate really well to things that they have trouble envisioning affect them personally. So that is the root of our hope with COVID-19. It is also the root of our political problems with COVID-19. Yes. Okay. Now, I'm not sure we're going to get through all of this today for like the first time. Probably should have started this last time rather than ask for questions. But the next to last thing I want to talk about today is parasite virulence and the transmission barrier, and look at ways in which these things interact. So there are 4 things that we have discovered that really bring home the importance of that transmission barrier. First is the difference in virulence between vertically and horizontally transmitted parasites. Second is the difference in virulence between vector-borne and non-vector-borne diseases. Third is the effect of co-infection, as Laura was asking about with strain 19 and strain 17. And the final one is the demonstrated effect of serial passage that enhances virulence in the laboratory. So first, vertical versus horizontal transmission. Horizontal transmission is diseases passed from one individual to another within a generation. So that's like Dwayne infecting me with something. Vertical transmission is being passed from parent to offspring. Okay. And generally, vertical transmission can occur during fetal development in humans. So that means that a vertically transmitted disease must allow the host to live long enough to reproduce. Well, that means they have to be less virulent. Vertically transmitted diseases are less virulent than horizontally transmitted diseases because they have to let the host survive long enough to reproduce. Tobacco mosaic virus in barley was a really very cool demonstration of this because it's vertically transmitted and there's clear selection for reduced virulence. And so I put it on there for your edification. I know you'll all read it. Okay, second, the distinction between vector-borne and non-vector-borne diseases. A vector is a third party that carries a parasite from one host to another. So that lowers the transmission barrier because you don't now have to rely on an infected individual coming into contact with an uninfected one. An infected individual can be in a hut in the rainforest, and a vector like a mosquito can carry that person's infection 20 kilometers over the rainforest to another individual. So vector-borne diseases like malaria are on average more virulent than non-vector-borne disease. The transmission barrier is lower because you have this third party. Third, what happens if you do have co-infection of, say, two different strains of a disease simultaneously in a single individual? Well, okay, so in that case, then you have two different genetic lineages competing for the resources of the host. And it's obvious, natural selection is going to favor the one that's most efficient. And that will generate higher virulence. Now, we didn't really understand how this worked until about the 1950s when we started deliberately doing experiments where we had more than one strain of parasite injected into human cell lines, for instance, and found that the more virulent one was the one that always won. Duh, because natural selection is selecting for the one that's most efficient, the one that's most virulent. So when you get co-infection, this competition between the differing strains makes the host much sicker than if it was a single genetic lineage. Okay, finally, serial passage. Serial passage is something that we do in the laboratory when we're trying to culture a parasite, especially a disease organism that we're culturing in cell culture. We directly take infective agents from one individual and inject them into another. Well, duh. Of course, there is no transmission barrier whatsoever then. So over time, cell cultures that are infected with parasites in the laboratory gradually develop much more dangerous, much more virulent parasites because there is essentially no transmission barrier. So strains of disease organisms that we keep in the laboratory can be more virulent than wild strains. Okay, I'm going to stop here and leave the last topic for Wednesday. Okay, and that's complex life cycles. So that's all we're going to do today. If anybody's in the room and wants an exam, please come to the front. If there's any questions, we'll take them now, but if not, we are done. Hey guys, we'll see you.